Hello everyone. Today we'll talk about a maternal imprinting pedigree problem which came in CSIR UGC in December 2025. Sorry, June 2025. So before discussing this problem, I thought to discuss about genomic imprinting a bit. What is genomic imprinting and how this life cycle of this imprinted gene works. So in case of genomic imprinting, what happens is, some cases, so we know that we, so in diploid organisms, there will be two copies of our gene. So sometimes what happens is in the sexually reproducing organism, during gametogenesis, one of the copy in one parent, for example, either father or mother, will be methylated and henceforth it will be silenced. For example, in this case of maternal imprinting, what is happening is the copy which is being passed on from the mother is being methylated. So basically, this is, let's say this is the gene location where we are talking about, okay, this is the, let's say this is the gene X, for example, where, so in this gene X, if this is methylated, so the, if this is methylated, then what will happen is this won't undergo any transcription at all. And so this copy will be non-functional. This copy will be rendered non-functional completely. However, in case of the paternal copy, this copy, as it has zero methylation mark, it will be completely functional. It will undergo transcription and this will produce the gene X. In case of paternal imprinting, however, the opposite will happen. That means this won't be happening at all. The paternal copy won't undergo transcription because of the methylation mark, and the maternal copy will produce the uh, gene X. So in this case, if something goes wrong with the other copy also, for example, this is the recessive mutated copy, or here if is the recessive mutated copy, for example, this is one, this is a full, like, normal uh, wild type copy. However, if let's say this is methylated and the other one is mutated, so this will lead to some sort of disorder. Now, how these methylation marks get passed on to the next generation? So suppose we are talking about two genes which is A and B, and these A and B genes are maternally and paternally imprinted respectively. So this one is paternally imprinted. So in that case, what will happen? So these two genes will pass on, and so here what is happening is one methylated copy is coming from the father mother for the A gene. However, the other one is completely fine. And let's say here also one methylated copy is coming from the father. Now, during, so let's say there are two offspring which has the same genotype of these. One is female, another one is male. What will happen that when they will produce gamete. So during the gametogenesis process, however, the somatic cells retain all these methylation marks here. During gametogenesis, these methylation marks will completely go away and the old imprints will be erased. And but as these genes are maternally and paternally imprinted respectively during gametogenesis, new imprints will arise. So the maternally imprinted genes again will be maternally imprinted and passed on to the next generation, and the paternally imprinted one will be paternally imprinted and passed on to the next generation. So how can we utilize this information to solve this question? So here what is the what they have asked us to study this inheritance pattern of gene X. A loss of function mutation in the gene will lead to a disorder in adult. That means if the genotype is, for example, this, where we denote the small x as the mutated copy. So mm, both copy of mutation, like both mutated copy, will lead to a disorder. Or we can say if there is a functional copy but that is also methylated, for example, and other one is mutated, that can also lead to the disorder. Further, the gene X is being imprinted, leading to its inactivation. Individuals who carry this mutant allele are marked with dot. So if there is a, a single mutated copy is present, that will be denoted as a dot, like this. While the individuals who show the disorder are shaded like this. Here we can see that the individuals are having this shaded um, phenotype. They are marked with shaded. So here what we can expect that either their genotype is like this or they have this genotype like this, but these are methylated. These are methylated completely. Now how to figure out if these are like which is the correct genotype. So we can see that the mother is having one mutated copy. The father is having zero mutated copy. So we can say this is like this. So we can definitely say one copy is coming from the father. And as we can say, as we can see that this is having these two individuals, 37 and 38, is having disorder. So we can say that genotype is like this, where the paternal copy is having the imprinting methylation mark, but and rendering it non-functional, and the maternal copy is recessive. So it at the end of the day leads to no functional copy. There won't be any functional copy, and they will undergo a disorder. Now, for this individual, what I can say is, so there is one mutated copy, and this mutated copy is definitely coming from the father, and the other individual is this. So we can say that this individual will have this genotype. Now this individual, so now again what we have to see that we have to, so the first option G next is maternally imprinted, it is wrong, it should be paternally imprinted. And the individual 3-6 carries an imprinted allele. Yes, definitely it should carry, because like others, any of this copy will be methylated completely. And because of that, let's the paternal copy, let's say this one, and this one is being methylated. So the statement B should be correct. Now, how to look for the genotype of this individual, whether this individual is like this or whether this individual is again like this. Now as we can say that okay, this individual is not having any disorder, so definitely it should have this genotype of this. And the father is also might be having a methylation mark from start, which is in the recessive copy, but it doesn't matter. Now, next we can say that okay, while gametogenesis, this mutated copy, this mutated copy is being passed here, and definitely it is being methylated. And here the maternal copy won't have any methylation mark at all. Here this individual will have this genotype because we know there is no mutated copy, and the mother here won't have methylation mark for sure. Now the father will pass on these two copies. Now we can say that all of these individuals, though they are not having any disorder, implying the methylation mark might be present here in the mutated copy itself, and the mother is only giving us the functional copy which has no methylation mark at all. Now we have to look for the offspring of the individual 3, 4 and 3, 5. If they have a child, what is the probability they carry one mutant allele? Is this, or what is the probability they will show a disorder? Is this? Which one is correct? So now we have this 3, 4 individual, 3, 4, and which and the 3, 5 individual which has this genotype like this. If they uh, if they have a child, then what is the possibility it will have a mutant allele? First of all, what is the possibility to get a mutant allele in this case? So there are three chances: either they will have this genotype, or they will have this genotype, the heterozygous combination, or they will have this genotype. So chance of having this gamete is half, and again here it will be half. So in this particular case, what will happen is, so in this particular case, this will be half into half plus, and also we know the ratio will be 1 is to 2 is to 1 here like this. So here the chance of getting this mutant allele is this, which is 2 into 3 into half, which is 0.75. So we can say that the chance of getting one mutant allele is 0.75. And why this the last option is incorrect is because, say for example, now the father can so the father, for example, any of this is the father, 3, 5 is the father, right? So now this can give us either like this methylation mark, or here this X copy is either coming from, so this can be either like this, or this can be either like this, and here this will be like this. Now the chance of having disorder is, so this chance of having this is 0.25, and this is also 0.25. So all total the chance will be 0.5 only, and not 0.75. So the last statement is incorrect.